शीतला सस्यश्यामला मातरम् वन्दे मातरम् शुभ्रज्योत्स्ना पुलकितयामिनी पुल्लकुसुमित द्रुमदलशोभिनी सुहासिनी सुमधुरभाषिणी सुखदां वरदां मातरम् वन्दे मातरम् वन्दे मातरम्.
Good morning, students. How are you? I think you all are doing well. Yes. Now we will going to learn about a new topic, that is story of microorganisms. What is that? Story of microorganisms, part 1. Part 2. Before entering into this topic, I want to ask you some of the questions. Why do we add buttermilk to the lukewarm milk? Why do we add buttermilk to the lukewarm milk? Next. Why do spoil the cooked food when we keep 1 or 2 days? Why do it spoil? అంటే గోరువెచ్చని పాలకు మజ్జిగ చుక్క ఎందుకు కలుపుతాం? ఫస్ట్ క్వశ్చన్. సెకండ్ వన్, కుక్డ్ ఫుడ్ getting spoiled very soon. What is the reason? What is the reason? For such kind of questions, find out the answers by so many scientists. It means so many scientists are working regarding such kind of questions. In that, the most important scientist, his name is called as Antony van Leeuwenhoek. Who is that person? Antony van Leeuwenhoek. "400 years back, this Antony van Leeuwenhoek, who researched about such kind of questions for find out answers. Who is that name? The person name is Antony van Leeuwenhoek. So, microbiology. Have you heard about this word? Microbiology. It is a branch of science. It is a branch of science. Science lo oka bhagam. What is microbiology? On this world, every living organism we can able to see through our naked eye. On this world, is it right? Every living organism we can see through our naked eye. Do you know one thing? Some of the living organisms which is around us, even though the living organisms are around us, but we are unable to see. We are unable to see. That is possible to means that living organisms can possible to see through only microscope. Such kind of organisms are called microorganisms. What is that? Microorganisms. That world is called microbial world. It means in our surrounding there is another world, that is microbial world. How can How can we know? Because of the Antonio Leeuwenhoek, we find out this microbial world. How? In the year of 1674, this microbial world came to exist. It means they found out by Antonio Leeuwenhoek. How he find out? He prepared one microscope, a single lens microscope. What is that? A single lens microscope, powerful microscope. With the help of that microscope, you found not only bacteria and also some other microorganisms also founded by this Antonio Leeuwenhoek. There are different types of microorganisms are present around us. మన చుట్టూ కూడా చాలా రకాల మైక్రోఆర్గానిజమ్స్ అనేవి ఉన్నాయి. But the first microorganisms are found out by Antonio Leeuwenhoek that moving and small moving organisms which is present in the water is called as animalcule. This animalcule is also called as, later it is called as bacteria. Not only bacteria and also some other microorganisms also you find out. So later he gave a name for them also. Okay. So according to that, there are different types of microorganisms. What is that? Different types of microorganisms are there. Microorganisms. What is that? Microorganisms. Some of the living organisms are we can able to see through our naked eye. But some of the living organisms are we can't able to see through our naked eye. Such kind of living organisms are called as microorganisms. What is that? Microorganisms. But how can we see? That can possible to see through only electron microscope or microscope. Okay. So in the microorganisms, these microorganisms are classified into 5 groups. How many groups? 5 groups. These microorganisms are classified into 5 groups. What are they? First one, bacteria. What is that? Bacteria. Second one, algae. Third one, fungi. Fourth one, protozoa. Fifth one, arthropoda. So likewise, the microorganisms are classified into 5 groups. So in the bacteria, how many types are there? See, Kochi bacteria, examples for bacteria examples. Kochi bacteria, Kochi. Bacillus bacteria and lactobacillus bacteria. These are the examples for bacteria. And come to the next, algae. What are the examples for algae? Algae, which is color in green. Algae means how it is color. What is the color of this microorganisms? Green in color. So what are the examples? Chlamydomonas. What is that? Chlamydomonas. Spirogyra. Diatom. What is that? Diatom. Spirulina. Edogonium and Creatum. These are the examples for algae. Come to the next. Fungi. Penicillium. Bread mold. Rhizopus and aspergillus. These are the examples for fungi. And protozoa examples. amoeba, paramecium, verticella. What is that? Verticella. These are the example of protozoa. Next, fifth one, arthropoda. What is this? Arthropoda. Example, cyclops, Danfia, scabious mite, eyelash mite. Cyclops, Danfia, scabious mite and eyelash mite. These are the examples for arthropoda. So, the microorganisms are classified into 5 groups. What are they? Bacteria, algae, fungi, protozoa and arthropoda. So like that, these microorganisms are classified. Okay, how can we see this microorganism? Is it possible to see? Yes, it's possible through microscope. Not with our naked eye, but it is possible to see through microscope. So for that, if you want to see microorganisms, we have to do an activity. We have to do an activity. So for that, what we have to do? Go to near the pond and collect some of the pond water. In that pond water, it should be some of the green saplings. It means green pieces. It should be there in that water. Okay, so that water we have to collect it from the pond, and in that pond water means what we collect in that water we have to take at least 1 drop of water and keep it on the slide. If you observe this slide with the help of microscope, we will find out different microorganisms in the slide. So when we are observing these microorganisms with the help of microscope, immediately we have to draw the diagram. While we are seeing, we have to draw the diagram what we are seeing in the slide. Then all the microorganism diagram we have to draw in our notebook while we are seeing. Okay, then after that we have to compare this diagram. With your textbook diagrams. What I explain here. Some of the diagrams are coincident, means it matches. Then immediately you have to write down the names. When we compare this both diagrams, means what we draw and what diagrams are in the textbook they are given. That both, if you match it, when we find the relations, immediately you have to write their names. It may bacteria, it may fungus, it may algae, it may arthropoda, whatever it may be, you have to write down their names. Check it and write down the names. It means in that water, so many microorganisms are there. So many microorganisms are there. Now, we will observe fungi. How can we observe fungi? What are the examples for fungi? Next, Rhizopus, Aspergillus, bread mold. These all are the examples for fungi. But how can we observe this fungi? With the help of microscope. Have you seen some of the umbrella-like structures? Wet rodent, wood plants. When we observe wet rodent, wood plants on the edge of this wood plants, we will see the umbrella-like structure. What are they? Mushrooms. What are they? Mushrooms. Mushrooms are one of the fungi, but this is not a microorganism. We can able to see. We can able to see. But not only that, I am not talking about that. When we are observing some of the bark, tree barks, on the tree bark, some of the white patches during the winter season, during the rainy season, we will observe some of the white spot on the bark, tree bark. Do you know what is the reason for that white patches? Fungi. What is that? Fungi. White patches. White patches on the bark. And also some of the umbrella-like structures also we will find out on the rotten wooden plant. They are called fungi. How can we see this fungi? If you want to see fungi, for that we have to do an activity. What is that? Take some of the rotten vegetables or spoiled bread. Spoiled bread. Anyone we have to select it for doing this experiment. For find out fungi. So when we are taking spoiled bread, from that we have to take little piece with the help of forceps. Forceps means two angles ఉంటాయి కదా పట్టుకోవడానికి two angles తో పట్టుకొని తీస్తాం. That is called forceps. Forceps తో మనం spoiled bread మీద నుంచి means బూజు అంటాం కదా. That one we have to take little amount from that bread. And this little amount of bread piece we have to keep it on the slide. We have to keep it on the slide and put one drop of water. Then after that, on the top of this object we have to keep one cover slip. And cover చేసేదాన్ని cover slip అంటారు. Cover slip. After that, this total slide we have to keep it under the microscope. So when we observe this slide with the help of microscope, in that what we are seeing, the picture we have to draw in your notebook. In your notebook. After that, this diagram, what you draw, that diagram, you have to compare with the textbook diagram. Then immediately some of the textbook diagrams are coincident with this, with your drawing diagram. Is it right? It may rhizopus, it may penicillin, it may not be penicillin, I think. Why? Because we have selected bread mold. So that's the bread. So that's the reason. What you draw? Maybe bread mold or rhizopus. So what I explained is all the names belongs to fungi. Rhizopus, bread mold, aspergillus, penicillin. These all are belongs to fungi. But now what we find out? Bread mold we find out. Bread mold we find out. So likewise we will find out, we will observe the fungi. We will observe the fungi. Now, how can we observe bacteria? How can we observe bacteria? So if you want to observe bacteria, we need curd. We need curd or buttermilk. So, how can we do this activity? How can we do it? For every activity we need microscope. Without microscope we can't able to do these activities. Is it right? Yes. So curd, microscope, slide. So first we have to take slide. So on the slide we have to take one drop of buttermilk. So this drop of buttermilk, we have to keep it on the slide. So first take 1 or 2 drops of buttermilk on the slide. Next, heat the slide. And when we keep this buttermilk on the slide, means that buttermilk we should heat at least 3 to 4 seconds only. 3 to 4 seconds, just heat it, then take it out. Next, after the heat, we have to add, means for this buttermilk, after heating, we have to add a little amount of crystal violet, means that is one type of strain and wrong anamata. You add it. Master, why we have to add this crystal violet to this buttermilk అంటే మనం చూసేటువంటి స్లైడ్లో ఉన్న ఆబ్జెక్ట్ ఏదైతే ఉందో అది బాగా క్లియర్గా కనిపిస్తుంది. సో దట్స్ ద రీజన్ వీ హ్యావ్ టు యాడ్ ద క్రిస్టల్ వైలెట్. అంటే ఏంటమ్మా, ఇట్స్ ఏ స్ట్రైన్ అంటే ఒక రంగు అనమాట. దెన్ ఆఫ్టర్ యాడింగ్ దిస్ క్రిస్టల్ వైలెట్, లీవ్ ది స్లైడ్ అట్ లీస్ట్ 30 to 60 seconds. లీవ్ ద స్లైడ్ at least 30 to 60 seconds. Don't touch it. After 30 to 60 seconds, we have to wash this slide. Wash ఏమని చెప్పేసి ఎలా సబ్బుతో పాల్పేయడం కాదు, జస్ట్ జెంట్లీ వీ హ్యావ్ టు వాష్. అంటే జస్ట్ పైపైన అలాగా వై వీ హ్యావ్ టు వాష్ ఇట్ మీన్స్ ఎక్స్ట్రా స్ట్రైన్ ఉంటే కనుక అది పోవాలన్నమాట. ఎక్స్ట్రా స్ట్రైన్ ఏదంటే పోవాలి. సో దట్స్ ద రీజన్ ఆఫ్టర్ 30 to 60 seconds we have to wash this slide gently. After washing this slide we have to keep it under the microscope. When we observe with the help of microscope that slide, we will find out a streak-like structures. పుల్ల ముక్కలు లాంటి నిర్మాణాలు మనకు కనబడతాయి స్లైడ్ మీద. These streak-like structures are called bacteria. Whatever bacteria it is, lactobacillus bacteria. What is that? Lactobacillus bacteria. When we are observing, during the observing, we have to draw the diagram. The diagram we have to compare with the textbook diagram. Then it matches to the lactobacillus bacteria. It matches to the lactobacillus bacteria. So likewise, we will observe bacteria with the help of microscope. With the help of microscope. Now observing algae. So for this we need pond water. Actually, all the algae are not microscopic. Chara, Spirogyra, these all we can see through our naked eye. But some of the organisms means algae we can't able to see through our naked eye. So for them we have to do this experiment. So how can we do it? Take pond water. In that pond water, some of the green saplings it should be. Means some of the green pieces it should be. So we will collect it. Then take 1 drop of water. We have to keep it on the slide. From the plant, what we collected, that water drop we have to keep it on the slide. Then cover the cover slip, keep it under the microscope. What we find out? We will find some of the microorganisms which belongs to algae. Okay, next some of the protozoa. How can we observe protozoas? If you want to observe protozoa, take little amount of hay. Hay means indicator. Then you have to keep this hay in the water. It means this hay soak in the water at least 3 to 4 days. Hay అంటే indicator. దాన్ని నీటిలో ఏం చేయాలి, మనం నానబెట్టాలి అట్లీస్ట్ 3 to 4 days. After 3 to 4 days, some of the decoction we will find. Decoction means brownish color water. So from that water we have to take one drop of water and keep it on the slide and covers the cover slip. Then we will observe with microscope. What we find out? In that water, some of the protozoa we will find out. Some of the amoeba or paramecium, verticilla, whatever it may be. Some of the protozoa we will find in that water. Like that, we are observing protozoa. Observing protozoa. So actually, in this way, we will find the different types of, means we will observing different types of microorganisms. Maybe algae, maybe bacteria, maybe fungi, maybe protozoa, maybe arthropods. Observing soil microorganisms. What is that? Soil microorganisms. How can you observe microorganisms from the soil? So for that, we need some soil from the matti and beaker with water. Beaker with water. Take a beaker. In that beaker, we have to pour water. In that water, we have to pour the soil and stir it. Stir it. Leave it some time for settle down the particles. And the matti kanalani kuda kalisi ala settle avvalani. Okay? And the water soil both are mixed together. That is called stir it. Then leave it for some time. After some time, we have to take one drop of water, keep it on the slide, this water drop, keep it on the slide, covers the cover slip, then we have to keep it under the microscope, then we will observe some of the microorganisms which is present in the soil. It means in the soil also there are some microorganisms. Microorganisms are present in everywhere. Where it may water, they can live on water, air, soil, and also bodies. Maybe it is animal body or plant body, and ice. Too much cold also they can able to live. Too much hot springs also. In the hot springs also they can able to live. Hot springs and also some of the desert area. Desert area also these microorganisms can able to live. It means these microorganisms are present in everywhere. These microorganisms are present in everywhere, like a God. So here, actually, some of the microorganisms, they can't able to live independently. And our microorganisms, they can live in human body or plant body as a parasite. These microorganisms all are cannot alive as independent. Some of them they are depending on others. Means they can live on other bodies. Such kind of microorganisms are called as parasite. అంటే ఆహారాన్ని అదంతా సంపాదించుకోలేదు, వేరే వాళ్ళలో ఉండి వాళ్ళ ఆహారాన్ని తింటా ఉంటాయి అన్నమాట. సో వాటిని ఏమంటామంటే పారాసైట్ అని ఏమంటామంటే పారాసైట్ నేచర్, యాజ్ ఎ పారాసైట్. Here, some of the diseases, even though some of the uses are there with the help of microorganisms, and also some of the harmful effects also is there with the help of microorganisms. some of the diseases caused by microorganisms. What are they? For example, TB means tuberculosis, typhoid, septicemia. Septicemia means blood poison. Septicemia means blood poison. These three diseases caused by bacteria. Bacteria means one type of microorganism. Next, skin diseases. Actually, skin diseases are caused by fungi. And also some of the microorganisms also caused for these skin diseases. And also some of the bacteria are also caused by the skin diseases. Means microorganisms, fungi, bacteria are caused for this skin disease. Next, malaria, amoebiasis. Malaria, amoebiasis. Amoebiasis means Jigata virochana. For these most diseases caused by protozoa. What is that? Protozoa. It means microorganisms, all the microorganisms are not useful to us. Some of the microorganisms are caused for different types of diseases. It means some of the microorganisms are harmful. According to that, the microorganisms are categorized into two groups: useful microorganisms and harmful microorganisms. These useful microorganisms and harmful microorganisms we will discuss in the next chapter. It means the microorganisms chapter 2. Okay? Thank you.